Okay, so this is the first question which I have taken. Okay, so this is question number 15, which has appeared in 2020 biochemistry section of GATE Life Sciences. So here the question has been asked that the polypeptides are either biosynthesized on the ribosomes using an mRNA template or chemically synthesized by Merrifields solid phase method. The correct direction of the peptide synthesis are. Okay, so how does this protein uh, translation protein synthesis takes place? So we know the central dogma. So in the central dogma, what we have, yeah, so we have the DNA that will get transcribed and give us the mRNA, messenger RNA, and then from this messenger RNA through translation process, we get our desired protein. Okay, so this is the central dogma. Okay, so from using this mRNA, so from the ribosome machinery, we'll get the polypeptide synthesized. Okay, polypeptide synthesis takes place, elongation of the chain takes place, elongation of amino acid chain. So in the ribosome, what is the direction of protein synthesis? So it has been asked C to N or N to C direction. So can you tell what is this N and C referred here? What is N and what is C in the question? Yeah, nitrogen and carbon. But in amino acid reference, what does that mean? Amino acid has all, right? So if you'll see the basic structure of amino acid, this is the carboxy terminal, hydrogen, and the R group, right? So this is the basic structure. So how? So they make a peptide bond, right? So this is the amino terminal, then and then R1, then we get the peptide bond NH, and then another amino acid will come. Okay, H group, R2, one R2, then again another peptide bond, and then another. So this is, suppose I made one. This is one tripeptide I made. Okay, three different R group, three different amino acid chains. So there are two peptide bonds they are joined with. So this side the amino end is free of one amino acid, and this side the carboxyl group is free of the amino acid. So this is the polypeptide chain where one side is amino terminal that is referred with N, and another side is carboxy terminal that is referred with C. So in ribosome machinery, the amino acid gets incorporated in this manner only. First this, and then this, and then this. So amino acid synthesis or the polypeptide synthesis takes place from N to C direction in the ribosome machinery. N, C, N, C. So this is the direction of the polypeptide synthesis using the ribosome machinery. One by one amino acids are being added and makes the peptide bond. Okay, so that C and N refers to the free amino terminal and the carboxy terminal. Okay, and accordingly it has been asked what is the direction of the synthesis. Okay, so in ribosome synthesis, the polypeptide synthesis takes place in N to C direction. So here out of four options, if we'll see, so this is C to N direction in both cases. So it won't be correct because for ribosome, for this particular ribosome machinery, we are seeing N to C. This N to C is correct. But for other case, we don't know. Is N to C, this could also be correct. And this particular option where C to N direction for the ribosome is incorrect. Okay, so out of four options, now you are left with two options. That is that for sure that for ribosome the synthesis takes place from N to C direction. Now the question comes in Merrifield solid phase method, what is happening? So Merrifield solid phase method, so this particular system takes place inside a um, this particular system takes place inside the live cell. Okay, so inside the live cell, using live cell or living organism, using ribosome machinery. Okay, using ribosome machinery, if the polypeptide synthesis takes place, that is from N to C direction, that is the polypeptide synthesis direction. But in cell-free translation system, in cell-free translation, where you break the cell membrane, take all the components, and then you try to synthesize, or artificially if you are trying to synthesize, artificial protein synthesis if you want to do artificial protein synthesis, so there you provide amino acid and it will make bonds with each other, the peptide bonds. So what is the direction of that? So for that we have one solid phase. Okay, so this solid phase, which is made of the styrene rings, so polystyrene polymer, so that is the solid phase. So, and that is having the chloromethyl group with that, that is the active group in the solid phase. So this solid phase is insoluble and easy to use for the artificial protein synthesis. So there, one by one, the amino acids are being provided and added. So first amino acid which is being provided here, carboxy terminal, and this is the amino terminal. So for artificial protein synthesis or Merrifield solid phase protein synthesis, this amino terminal is blocked. Okay, it is blocked. So this particular end is free. Okay, so here further to this solid phase, they make this ester bond. Okay, ester bond they make. So now comes up. So like this particular amino acid, okay, this particular amino acid gets attached to this solid phase, and here the amino terminal is blocked. Okay, so for acting the next amino acid, in the next step, they do the deprotection. So if this amino terminal is protected, and for adding the next amino acid, they first deprotect it. Okay, deprotected. So in the deprotection step, it gets free. Okay, it gets free. So very first is 1 second. Just give me 1 minute. Give me 1 minute. My pen is not working. So here they first, so after addition of the first amino acid, they deprotect the amino group of this firstly added amino acid, and after it gets freed, So after it gets free, you have the amino terminal free. You have the first amino acid added with the ester bond to this particular solid phase. Okay, so now after the deprotection, they again bring one another amino acid whose end terminal is also protected. This amino terminal is also protected, but the C terminal is free. So now this amino acid will be added here, and peptide bond is formed. So that is how another amino acid gets added. So first amino acid, then the second amino acid will be added, and now in order to get the other amino acid added, same thing will be repeated again the deprotection and addition of another protected amino terminal protected amino acid, and then the synthesis will continue, and at the last step they will remove the this resin or the solid phase, and we have the artificially synthesized amino acid ready. So here if you will see the synthesis which is taking place, it is from the C terminal, sorry, from the C terminal to the N terminal. So that is why the direction of this particular synthesis is C to N direction, while in ribosome synthesis, the direction of amino acid synthesis is N to C direction. So that is why the answer of our question will be, so N to C direction on the ribosome, while C to N direction on the solid phase. So B is the correct option and C is the wrong option here. And this is my NPTEL reference. Thank you.